Hello everyone, so this is the third video in the series of lipid metabolism or beta oxidation. So we have already talked about beta oxidation in the previous two videos, alpha oxidation, omega oxidation, and the production of ketone bodies and the related pathways. So in this video we are going to talk about the anabolism or the production of fatty acid, how lipid biosynthesis occurs, that we will talk about in this video, and how that process is regulated. So let's see step by step. So the absolute cost of constructing a palmitate requires massive energy and energetic and reducing power. So a palmitate has 16 carbons, and to produce the 16 carbon fatty acid, what we require is 8 acetyl CoA molecules, 7 molecules of ATP, and 14 H plus ions, and 14 NADPH. NADPH is being used for reduction. This is a reducing agent. Then we require ATP for the 7 cycles of production of palmitate, since 8 acetyl CoA will be added into a 16 carbon molecule through 7 cycles. Each cycle will add around 2 carbon, while the last step will add 4. So that is that. Then output will be 1 molecule of palmitate, 8 coenzyme A will be released, 7 ADP after from ATP will be produced, 7 pi, and 6 H2O molecules. So the direct rule of 16 carbon in palmitate, only 2 comes directly from acetyl CoA, the remaining 14 are donated from malonyl CoA. So this is a point to remember, that the 16 carbons of the palmitate, only 2 comes directly from acetyl CoA, the rest 14 are donated by malonyl CoA. Now malonyl CoA is also produced through acetyl CoA, that is another thing. So this is the whole reaction. Let's see step by step how palmitate is being produced. So the inner mitochondrial membrane creates strict physical barrier of acetyl CoA. Now you must remember that after the glycolysis, now the acetyl CoA through beta oxidation had occurred inside the mitochondrial matrix. So the acetyl CoA is actually, through beta oxidation, acetyl CoA is being produced, and acetyl CoA is inside mitochondrial matrix. But the biosynthesis of lipid molecules or fatty acid actually occurs in cytosol. So this acetyl CoA needs to go inside the cytosol from mitochondrial matrix. Acetyl CoA is impermeable. This inner mitochondrial membrane doesn't let acetyl CoA pass. So what happens is that one acetyl CoA molecules forms a combination with oxaloacetate. It reacts with oxaloacetate and forms citrate. This molecule, this is a TCA cycle molecule, and this citrate is actually is permeable through this inner mitochondrial membrane and comes out inside the cytosol. The citrate can come out into the cytosol. Citrate is freely permeable. It acts as a carbon Trojan horse, carrying the vital acetyl group into the cytosolic synthesis machinery. Once inside the cytosol, it will once again be converted into 1 acetyl CoA and a oxaloacetate. Right? So that is the process, how acetyl CoA comes out into the cytosol, because the biosynthesis actually occurs inside the cytosol. So this is the whole process in detail, how the citrate malate shuttle relocates carbon while actively generating reducing equivalents. Let's see. So acetyl CoA is inside the mitochondrial matrix. Step 1 is acetyl CoA is added to oxaloacetate to produce citrate. Citrate crosses the membrane. So acetyl CoA added to oxaloacetate produces citrate, that is then sent to cytosol. Now step 2 is ATP citrate lyase cleaves citrate back into acetyl CoA and oxaloacetate costs 1 ATP. Citrate is broken down acetyl CoA and oxaloacetate through the enzyme named as ATP citrate lyase. This breaks down citrate. The next is that this acetyl CoA then moves forward for the biosynthesis of lipids, while the oxaloacetate will now need to go back inside the mitochondrial matrix again for the TCA cycle, or for this again for the same cycle. So the step 3 in the cytosol is malate dehydrogenase converts oxaloacetate into malate, consumes 1 NADH. So oxaloacetate is actually converted into malate, and this malate is again permeable of the inner mitochondrial membrane and can move inside the mitochondrial matrix. This malate then produces pyruvate. Pyruvate carboxylase converts pyruvate back to oxaloacetate. So here once again oxaloacetate is produced through pyruvate. So this is the whole cycle, and it generates 1 NADPH and CO2 per cycle. Now acetyl CoA carboxylase act as the irreversible committed step for lipid biosynthesis. So once acetyl CoA is now inside the cytosol, it is now inside the cytosol. So inside the cytosol, what happens is that acetyl CoA undergoes carboxylation through an enzyme acetyl CoA carboxylase. Acetyl CoA undergoes carboxylation. That is, a carbon is added through carbon dioxide or carbonate molecule, and this produces acetyl CoA undergoes carboxylation, produces malonyl CoA, and the enzyme used here is acetyl CoA carboxylase ACC. This is the first step. The carboxylation of acetyl CoA is the first step in the lipid biosynthesis. This acetyl CoA carboxylase requires biotin as a prosthetic group for acting. So the ultimate gatekeeper, because this reaction is completely irreversible and dictates a continuous flux of carbons into fat storage. Now let's move on. This is the whole process of biosynthesis. So we already know acetyl CoA has produced malonyl CoA through the carboxylation through the enzyme ACC. This has already occurred. Now the process is as follows. The step 1 is condensation. Acyl malonyl CoA ACP condensing enzyme merges substrate into acetyl ACP, acetoacetyl ACP, releasing CO2 and ACP. So this enzyme, acyl malonyl ACP condensing enzyme, what it does is that it uses a acetyl ACP and a malonyl ACP produced through the carboxylation process and merges them. A condensation reaction happens and the two molecules are merged to produce acetoacetyl. So acetyl ACP and malonyl ACP has condensed to form acetoacetyl ACP. This is the first condensation reaction. The step 2 is reduction. So acetoacetyl ACP then undergoes reduction. For the reducing agent is again NADPH. This is the reducing agent. It provides H plus ion. So D3-hydroxybutyryl ACP is the molecule that is produced after acetoacetyl ACP is reduced. The enzyme used here is beta ketoacyl ACP reductase, and it utilizes NADPH as reducing agent. So acetoacetyl ACP produces D3-hydroxybutyryl ACP through reduction. Then comes the third step. The third step is dehydration. The first was condensation, then reduction, then dehydration. So now the 3-hydroxybutyryl ACP is converted into 3-D3-hydroxybutyryl ACP is dehydrated. 1H2 molecule is released to produce crotonyl ACP through dehydration. Now comes the second reduction step. The second step was the reduction, and again the fourth step is reduction. The first was condensation, then reduction happened, then dehydration, then again reduction. So crotonyl ACP utilizes NADPH as a reducing agent to reduce it into butyryl ACP. Enoyl ACP reductase utilizes a second NADPH to form butyryl ACP. Now butyryl ACP, the butyryl ACP had been produced, and this enters the next cycle, right? And this cycle goes on and on and on. Every time malonyl ACP adds 2 carbon into the cycle, and every time the cycle goes on, the extra 2 carbon has been added into the molecule. So this is how 16 carbon palmitoyl ACP is reached. So the cycle repeats, adding 2 carbon from malonyl CoA in each turn until the 16 carbon palmitoyl ACP has been produced inside the cytosol. This palmitoyl ACP with 16 carbon is the last step, is the end product in cytosol, right? So cytosol does not have the mechanism to produce a carbon compound more than a fatty acid more than 16 carbon. The rest of the process will happen in smooth endoplasmic reticulum. So let's move on to the next slide. So the smooth endoplasmic reticulum moves palmitate to fulfill diverse biological requirements. So there are two processes that occurs. First is elongation, that is after 16 carbon palmitate has been produced, but the body requires more carbon, fatty acid with more number of carbons, for example 20 carbon arachidonic acid, or there are also fatty acid with 36 carbons. For their production it could not occur in cytosol. So the elongation procedure now occurs in microsomes or smooth endoplasmic reticulum. Microsomes are actually small pockets made in smooth endoplasmic reticulum. So elongation occurs in microsomes, occurs in SCR, uses malonyl CoA as the 2 carbon donor and NADPH for reduction, operating on CoA derivatives rather than ACP. Now here is another important fact, that until this point of palmitate, ACP was the carrier protein, that acyl carrier protein was being used as the carrier molecule. Now in microsomes or in smooth endoplasmic reticulum, once again ACP will be removed and CoA, coenzyme A, will be added for the molecule to function rather than ACP. So in each cycle, 2 carbon donor of malonyl CoA will add 2 carbon in each cycle to let the elongation of palmitate, right? Then there is a second process called desaturation. So desaturation is the process of inserting a double or triple bond inside the fatty acid chain. So as you can see, the palmitate has 16 carbon and it has 0 double bonds. There are no double bonds in palmitate. And again, this insertion of double bond can only occur in smooth endoplasmic reticulum. It cannot occur in cytosol. The cytosol doesn't have enzymes for that. So what are the enzymes that actually we require for insertion of a double or triple bond in a fatty acid? So they are NADH, cytochrome B5 reductase, cytochrome B5 and desaturase. These are the three enzymes required for inserting a double or triple bond in a fatty acid chain or desaturating them. So what happens is, there is a saturated fatty acyl CoA. We require NADH, H plus O2, and a mono unsaturated fatty saturated acyl CoA is produced, NAD plus and 2H2O. Now it is important here to note that the mammalian structure or the mammalian body has some limit. Mammalians lack enzymes to insert double bonds beyond carbon 9. For example, so this is carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Right, so this is a fatty acid chain with 12 carbons, but mammalian structure cannot insert a double bond in more than this. After this carbon, they could not insert a double bond. Right, so there are some important fatty acid like linolate, which has cis delta 9 and dental 12, that is double bond on 9th carbon as well as 12th carbon, that cannot be produced in the body, as well as linolenate, all cis delta 9, delta 12 and delta 15 with double bonds at 9, 12th and 15th position. These are something that cannot be produced inside the body. This cannot be produced in the body. So they become essential dietary fatty acid. You have to take them in your diet, because they become the precursor molecules for the production of a lot of fatty acid that has double bond or triple bond after the carbon 9 that cannot be produced in the body. So these have to be taken through diet for the production of lots of other molecules. So this is a overall process of fatty acyl CoA and how that produces triacyl glycerols. How triacyl glycerols are produced. So what we require is the production of phosphatidic acid. So there is a glycerol backbone, which means there is CH2OH, CHOH and CH2OH. This is a called glycerol. This is glycerol backbone, as you can see here. And an enzyme called phosphatidic acid synthase, it leads to the production of phosphatidic acid through this molecule. Once phosphatidic acid has been produced, what it does is that it undergoes a reaction to produce diacyl glycerols by the addition of two acyl CoA. For the production of diacyl glycerols, what the diacyl glycerol will mean is that this OH and this OH has been removed, and an acyl group has been added. In these two positions. So this is now DAG or diacyl glycerols. There are two acyl groups in the glycerol backbone. So this is DAG, diacyl glycerol. How this is produced? There is a glycerol molecule and two acyl groups come bind to this. So diacyl glycerol has been produced from phosphatidic acid, and after diacyl glycerol, another acyl group will replace this and will finally produce triacyl glycerol, tag. This is how triacyl glycerols are produced. Now the regulation of this production of lipid biosynthesis, how this is regulated. There is no process in the body that occurs all the time. All the process that occurs in a body needs to be regulated when something happens and when something is stopped. So that we will see in this slide. So ACC is, we have already studied about ACC. If you do not remember, I'll show you. This was ACC. It was what it does is that it converts acetyl CoA into malonyl CoA by carbozylation. Through carbozylation of acetyl CoA, malonyl CoA is produced, and this is the first step. This enzyme leads to the first step of fatty acid synthesis. This is the first step in fatty acid synthesis. So this enzyme is really important in lipid biosynthesis. Now let's come back. So ACC is important for lipid production. Now there are another two molecules. There is insulin and glucagon. Let's see their function. So let's say this is a blood vessel and these are the RBCs. This is a blood vessel and these are red blood cells, RBCs. So if you have taken food, you are in fed state, you have taken food, there will be glucose in blood. There will be glucose in blood, and when there is high level of glucose, insulin is a hormone produced through Langerhans cells in pancreas. Pancreas produces insulin. This insulin this is in high level of glucose in blood. So what insulin does is that it makes tissues take up this glucose from blood. As you may already know, in case of insulin deprivation leads to a condition called diabetes mellitus. In such patients, there is always a high level of glucose in blood. So that is that. Insulin, what it does, it makes tissues uptake glucose from blood, and this is produced when you have taken food, you have eaten food. Now there is another hormone with the opposite effect. Let's see that. Is called, in case you haven't eaten, you are fasting, you haven't taken any food, there is no glucose in blood, but we do require some amount of glucose in blood, right? So where would that glucose come from? The tissues will release glucose into the blood. So the opposite effect will happen when you are fasting. The tissues will release glucose into the blood, and for this mechanism a hormone called glucagon, this acts exactly opposite of insulin. This makes the tissue give up their glucose into the blood. So that is the whole process that one must remember. So what was happening here? When you have taken food, insulin has been produced, there is a lot of glucose in the body, and where will all that glucose go? So that glucose needs to be converted into fatty acid or lipid. So for that, insulin is being produced, and insulin acts in another way. It dephosphorylates, it removes the inactivated phosphorylation of ACC enzyme and makes this ACC active. Insulin dephosphorylates ACP and makes it active, and once ACC is active, it produces lipid. So when you have intaken food and there is a lot of glucose, that glucose needs to be converted to lipid, so ACC is activated through insulin. Insulin signaling stimulates dephosphorylation of ACC, activating it. Citrate is another molecule. Citrate is an intermediate of TCA cycle, signals high energy. So this citrate allosterically activates ACC. Now there is another part of the story, that is energy depleted state, which leads to inhibition. In case you haven't eaten, what if you haven't eaten anything? You are in fasting state. There is no ATP inside the body, there is no energy inside the body. For example, there is high AMP ATP ratio, there is high AMP and low ATP, which means there is low energy inside the body. In that case, AMPK, that is AMP dependent protein kinase, this is AMP dependent protein kinase molecules always phosphorylates other molecules. So in case of high AMP level, that is low energy, AMPK is activated, which leads to the phosphorylation of ACC, and once ACC is phosphorylated, lipid biosynthesis will stop. Phosphorylation of ACC leads to its deactivation. So in case there is no energy, there is no glucose to produce that will need to be stored in case of lipid, so lipid biosynthesis must stop. That will occur through AMPK phosphorylating ACC. Palmitoyl-CoA. So palmitoyl-CoA is the last end product of this biosynthesis. If there is very high level of end product of any reaction, if there is very high level of palmitoyl-CoA inside the cell or inside the body, then that will also activate AMPK, which will again phosphorylate ACP and deactivate it. So palmitoyl-CoA also inactivates ACC. Okay, so that was the whole story of regulation. Now there is also a reciprocal control mechanism. The product of active ACC is malonyl-CoA, specifically inhibits carnitine palmitoyl transferase 1 CTPI. This guarantees that newly synthesized fatty acids are blocked from entering the mitochondria for beta oxidation. Now here is the thing, your body worked so hard to produce the fatty acid through biosynthesis process. So much energy has been almost 7 ATP is being utilized for this production and stuff. So all this effort will go to waste if the produced palmitic acid instantly enters inside the mitochondria and gets beta oxidized. So the whole process will go to waste. So in order to stop that, malonyl-CoA, which is produced through ACC uses acetyl-CoA and produces malonyl-CoA. So this malonyl-CoA is actually the product of this ACC. This malonyl-CoA actually inhibits a protein called CPTI, which is carnitine palmitoyl transferase 1. This is the transfer molecule that lets fatty acid enter inside the mitochondria for its beta oxidation. So malonyl-CoA inhibits that molecule and does not let fatty acid enter inside to the mitochondria, so there cannot be any beta oxidation. So that was the regulation. Now there is also hormone sensitive lipase. mirrors ACP regulation to enforce reciprocal control. So there is also hormone sensitive regulation of lipase. So the fed override. When you have intaken food, when you have eaten food, insulin increases rapidly and insulin decreases cyclic AMP molecule, promoting the dephosphorylation and inactivation of HSL, hormone sensitive lipase, while simultaneously turning ACC back on. So insulin negatively affects cyclic AMP production, and cyclic AMP production is reduced. When cyclic AMP is reduced, then the inactivated hormone sensitive lipase is activated. Once hormone sensitive lipase is activated, so in case of insulin, this negatively affects cyclic AMP, and when cyclic AMP is negatively that is reduced, what it does, it inactivates the hormone sensitive lipase. Inactivation of hormone sensitive lipase occurs while simultaneously turning ACC back on, which will produce lipids. What hormone sensitive lipase does is that it breaks down lipids into, leads to its beta oxidation, produces acetyl CoA. So hormone sensitive lipase what it does, breaks down lipids. And when there is already insulin is being produced, when there is already a lot of glucose in the body, glucose is already high in the body, that's why insulin is being produced. So insulin negatively reduces the cyclic AMP molecule, because there is already glucose in the body. There is no need to break down further lipids through hormone sensitive lipase to produce more glucose, right? So this pathway is stopped through insulin. While on the other hand, glucagon and epinephrine, glucagon is produced when there is low glucose in the blood and more glucose is required. In that case, glucagon is being produced. So it positively affects cyclic AMP. Cyclic AMP is increased, so it activates hormone sensitive lipase, and once hormone sensitive lipase is increased, lipids are broken down into acetyl CoA at the end, which will produce energy. So that is what happens. This is when you are fasting, this is when you are fed, you have taken food. Epinephrine is fight or flight response, again you will need energy, so this is activated. So the starvation trigger, glucagon epinephrine raises cyclic AMP, activates CAM dependent protein kinase PKA, and PKA phosphorylates hormone sensitive lipase. Unlike ACC, phosphorylation activates hormone sensitive lipase. And once phosphorylated and activated hormone sensitive lipase hydrolyzes stored triacylglycerols into adipose tissue, flooding the blood with free fatty acid for peripheral tissue and energy use. Now, lipid homeostasis demand continuous macromolecular transit between liver and adipose tissue. So this is a hepatocyte cell of a liver. The export route in fed state is that it synthesizes palmitate, packages into triacylglycerols and exports as very low density lipoprotein VLDL. In liver, hepatocyte palmitase has been produced and packaged into triacylglycerol and then exported as VLDL, very low density lipoprotein. This VLDL leaves the hepatocyte of liver, is transported through the body and generally goes to adipocyte cell, the fat tissues, the adipocyte cells, and here extract fatty acid from VLDL for internal fat storage. The fat is taken out from VLDL and stored. Now the mobilization route, fasted state. This is when you have eaten, the fed state, right? So fat is being stored. In case you need mobilization, in case you need free fatty acids, what will happen then? So in that case, in the fasted state, active hormone sensitive lipase in the adipocyte releases free fatty acids. These complex with albumin for transit back to the liver and muscle for beta oxidation. So free fatty acids are produced in the adipocytes and they transit back to hepatocyte by combining with a protein called albumin. So this is the whole process. Now the last table that shows how it works. So fatty acid synthesis occurs in liver and adipose tissue. Subcellular site is inside the cytosol where biosynthesis occurs. Acyl carrier is acyl carrier protein ACP. Coenzyme required is NADPH as a reducing agent. Malonyl CoA is the 2-carbon compound that gives the carbon, that is the donor. The transport system used is citrate shuttle to move acetyl CoA from mitochondrial matrix into the cytosol. The key inhibitor is long chain acyl CoA, ACC, inhibits ACC, right? The hormonal promoter of fatty acid synthesis is high insulin. Beta oxidation, that is breakdown of lipids or fatty acid, occurs in muscle and liver. The subcellular site is mitochondria. Beta oxidation actually occurs in mitochondria. The acyl carrier is coenzyme A. The coenzyme requirement in this case is FAD and NAD+, which get reduced. Carbon units is acetyl CoA. And the transport system is carnitine shuttle that makes the molecule come inside the mitochondrial matrix. The acyl CoA enters in the mitochondria through carnitine shuttle. The key inhibitor is malonyl CoA that inhibits CPTI. We already talked about this. Malonyl CoA inhibits this molecule, this CPTI, that stops the fatty acyl from entering inside the mitochondria for beta oxidation. Hormonal promoter is glucagon. Glucagon inhibits beta oxidation. Glucagon will increase the beta oxidation. Yeah. So thank you for watching this video. I hope this helps. This was the last video in the series of lipid metabolism. We have already covered beta oxidation in previous 2 videos, and this was the last video talking about biosynthesis of lipid. Thank you.